Hello, everyone. Welcome to the second podcast for day 28, covering the second part of Objective 1. In this objective, we're going to talk about convergent evolution, fossil records, biogeography, and theory. Let's go ahead and get started. Up to this point, we've talked about homology. And in homology, we have similar structures arising from a common ancestor. So, for instance, we talked about humans, four arms, and say whale flippers. They all originate from this common ancestor. They descended from this common ancestor. And so we have a shared structures. Different functions, but a shared structure. I'm going to erase this, and I want to talk about something called convergent evolution. And this is when we have distantly related organisms with similar structures. So, for instance, we can't explain, in convergent evolution, we can't explain why these two organisms have a similar structure based upon a common ancestor. It would be as if you had these two common ancestors that are distantly related to each other. Way back here somewhere they shared a common ancestor. But we end up with a structure that is similar. And same here. That wasn't found in this common ancestor, and wasn't found in this common ancestor, and wasn't found in some distantly related common ancestor. So how is it we could get a similar structure? Okay, let's talk about an example from your book. Your book talks about marsupials from Australia and placental mammals from North America. It's important to remember in this example that they are both share lineage with a common mammal. I'm going to write that up here so you can see it. Common mammal. These are both mammals. But all of these marsupials have very special characteristics that make them marsupials, and all of these along this evolutionary line have special characteristics that make them placental mammals. Throughout the course of evolution, natural selection allowed for certain kinds of adaptations to occur, and many different kinds of marsupials arose. One of these marsupials is known as the sugar glider. And placental mammals here, throughout the evolution of placental mammals, natural selection allowed for the adaptation of many kinds of mammals. One such mammal found in North America is the flying squirrel. They both end up with some similar structures like these gliding wings, and they both have big eyes. These structures of a gliding wing and a big eye were not found in either the common ancestors or the very distantly related common mammal. So we can't say that these structures arose because of homology, because of something that a common ancestor had. They rose independently. So how could they rise independently? Well, they rose independently because both organisms had shared a common environmental pressure. There was some sort of adaptive advantage for the sugar glider and the flying squirrel to live in trees and hunt at night. This allowed, the gliding wings allowed them to swoop down upon their food or to get away from predators. The big eyes helped them hunt at night. So both of these features allowed them to survive, get away from prey, to hide, to find food, and to live at nighttime, or to have their active part of their life at nighttime. So we call this convergent evolution because similar features converge, gliding wings and big eyes, and we cannot explain them based upon a common ancestry. So I want to introduce one more term here. I want to introduce another term here, and this is analogous structure. And analogous structures are similar structures between different organisms that cannot be accounted for by a shared ancestry. analogous structures do not arise from a common ancestry. Rather, they independently arise, as we see here in Convergent Evolution. Okay, I'm going to erase this. Alright, your book now talks about the fossil record and how the fossil record can be used to show patterns of evolution. It can tell us how organisms of the past differ from present-day organisms, and how looking at those structures in the past organisms and comparing them to the present-day structures, we can see how they have slowly changed. And your book briefly mentions an example of the stickleback fish. And so we know this common ancestor of the stickleback fish, and the very earliest stickleback fish. We know it had a pelvis and functioning hind limbs. It could walk. It could use the pelvis. Throughout the course of evolution, we end up with the stickleback fish, which of course doesn't have hind limbs and also has a reduced pelvis. And it's certainly a non-functioning pelvis. And we can compare the fossil record over time, and what we discover is that the pelvis is slowly being reduced. If we look at the pelvis here in this fossil, compare it with this one, to this one, to this one, to our present day stickleback fish, we can see that the pelvis has slowly reduced in size because its function is no longer needed. So it's gotten smaller and smaller and smaller. This is a great example of two things. One, that natural selection certainly occurs, but it's a slow process. Sometimes there's a misconception. One day you had an organism that had legs and a functioning pelvis, and the next day you didn't. Or one generation you had it, the next generation you didn't. Evolution is a very slow process. And the fossil record shows how slow that is. From a large functioning pelvis to a reduced non-functioning pelvis. And what's interesting is that the second point I want to make is how molecular evidence helps support this story. There's this gene, and the gene is called PITX1. You can write it down if you want, but there's no reason to memorize it. This name of this gene will never appear on your exam. But it's an interesting explanation as to what's happening here. How do we lose a pelvis over time? Turns out that this gene in animals like humans who have a functional pelvis and hind limbs, this gene during embryonic development is highly expressed in the part of the embryo that makes the pelvis and the hind limbs. We know that over time in the ancestors that would then become the stickleback fish, that the expression of this gene goes from this to lower, and then finally to non-existence. The expression of this gene in the stickleback fish has been greatly reduced over time. Without a functional PITX1 gene, you cannot form the pelvis. And the reason it takes so long is because the expression didn't go from completely on to completely off. It had to gain enough mutations over time for the expression to get lower and lower and lower, to the point now where we don't see a functioning pelvis, and it's a very, very small pelvis. It is in itself a vestigial structure that we talk about. Alright, your book goes over some other examples of how the fossil record is used, but I want to move on to the last topic of biogeography. So let's write biogeography here. So what does biogeography mean? Biogeography means the geographic distribution of species. So that's the definition of biogeography, but we need to also talk about another term called continental drift. Continental drift is the slow movement of the Earth's land masses. Evidence supports that about 250 million years ago, there was a solid landmass formed. All the land masses collided and formed this big mass of land. And this mass of land was called Pangaea. Then we know about 200 million years ago, or I should say, this evidence suggests that 200 million years ago, we see this landmass breaking apart. And then about 20 million years ago, they ended up in approximately their current locations. Give or take 100 to 200 kilometers. So pretty close to where they're at now. We can use these data, this information, to understand a little bit more about how organisms evolve. I'm going to erase this colored in area here. We still have Pangea here. So if you had an array of organisms here spread throughout Pangea, so to speak, and when it broke up, you would have some of these organisms, maybe some in all of them, maybe not all of them. And we can see how being in these new environments, as continental drift continued to move them and to move them to new locations, over time, granted, look, we're talking about very long times, right? These changes allowed for specific adaptive evolutionary traits to appear, or to be selected for, I should say. Darwin noticed this when he was working along the coast of South America. He recognized that there were organisms in South America that resembled organisms he might have seen in Europe, but they were still quite different, that they were better suited for life in the areas of South America. But they had similar features to them. When he went to the Galapagos, he also saw this, that there were animals that were very similar to those in South America, but had adapted new traits on the island of the Galapagos. So by studying these different populations, biologists can learn a great deal about the adaptive evolution of the organisms that we see on Earth. That ends today's podcast. In this podcast, we talked about the different lines of scientific evidence that supports evolution. This support comes from many different disciplines, from geography to geology, molecular biology, genetics, archaeology, taxonomy. All of these lines of evidence support the theory of evolution. If you have any questions, please make sure you come see us, and we'll be happy to help you. Thank you. Bye.